Today we're starting on a new chapter that has to do with phenotypes that are not related to Mendelian genotypes. So up until now we've been talking about individuals that are sexually reproducing individuals that are getting half of their information, their genetic information, from one parent and half of their genetic information from another parent, and that combines to produce genotypes, and then the genotypes are expressed to produce phenotypes. And so what you get is you get Mendelian inheritance. Now in the last chapter we were looking at some different descriptions of how phenotypes kind of develop or express themselves based on not what the genotype is, but how those alleles become proteins. So what the protein actually does in the cell, and whether it can work as a true dominant, which means that the hybrid is dominant, or whether it acts something kind of different from that, like an incomplete dominant or co-dominant or hierarchical dominance or whatever we're talking about. Now in this chapter, we're going to be looking at some examples of inheritance patterns, and here we're talking about phenotypes that don't come from the individual's genotype in the way that we normally think of Mendelian inheritance. So here we're talking about maybe a genotype that doesn't cause a phenotype, or a phenotype that doesn't come from a genotype, or we're talking about, say, a different type of DNA that we've inherited. And this all makes sense as we start going through the different things. So this is all of the things that don't follow Mendel's rules. That's why it's called non-Mendelian inheritance. So we're no longer talking about the genotype-phenotype correlation in the individual. So here we don't use Mendel's first law, random segregation. We don't have to worry about homologs being passed down from parent to child. We don't have independent assortment, so we don't have to worry about multiple genes. We don't have to worry about dihybrid crosses or monohybrid crosses or trihybrid crosses at this point. So, in these examples, we're going to look at 3 different types of inheritance. In these examples, your phenotype of the individual might not reflect the genotype of that individual. Now, the first thing we're going to cover is something called maternal effect. And maternal effect obviously has to do with the mom. Maternal means mom. And in this case, it's not the offspring's genotype that causes the phenotype, but the mother's genotype that causes the phenotype in the offspring. And we'll look how that happens in a bit. We'll also kind of briefly go into something called epigenetic effects. And an epigenetic effect is something that happens in the chromatin that gets passed down, but it's not genetic. It's not in the gene itself, it's in the structure of the DNA. So the structure of how the chromosome is passed down. We'll see what that means in a bit. And then the third type is that the genotype may not be in the nucleus. So there are other types of DNA that get passed down from parent to child, and this is also called maternal inheritance because it's usually passed down in the egg. And so in this chapter, students kind of sometimes get confused between the maternal effect and what we call maternal inheritance. They're very, very, very different things. And specifically, we call this extranuclear inheritance, meaning that it comes from outside the nucleus. So, predicting what we're going to talk about, what other organelles in a eukaryotic cell have DNA besides the nucleus?
Mitochondria and chloroplast.
Mitochondria and chloroplast. So we're going to be talking about those toward the end of the chapter. Now, today we're going to be talking about snails first. And if you look at these two snails, how do they differ?
Larger tail.
One's larger than the other. How else do they differ? Well, the color's kind of similar. But I heard orientation, did you say? Yeah. So they point different directions. Does everybody see that? So if you were to put your hand up here with your thumb going forward, we would see that this snail curves this direction, but this snail does not. This snail actually curves this direction. So I'm using my left hand for this one, and my right hand for this one. So this is a right-handed snail, and this is a left-handed snail. Just like people are right-handed and left-handed, right? So the handedness, whether it's left or right, is not a genetic trait in the individual. It is a maternal effect, meaning that the phenotype of these two snails has nothing to do with their genotype, and 100% what their mother's genotype was when that egg was developing. And we'll go through that in a little bit more detail. So when we start talking about the maternal effect, we want to consider gametogenesis. So what is gametogenesis? We didn't really cover it in chapter 3 much. That's the production of gametes, right? So genesis means kind of the beginning or the creation. And so gametogenesis would be the creation of gametes. So when most mammals produce gametes, they produce eggs or they produce sperm based on what set of gonads they have. And so sperm, in comparison to eggs, are tiny. They are so small when you compare them to the size of an egg. In fact, this is an egg, and these are sperm. Very, very small in comparison. Now, hopefully that kind of really illustrates this idea that an egg is huge, but the nucleus of each one is the same size. They each contribute the same amount of genetic information in the chromosomes, right? Because they're each haploid. And so the sperm is really just a nucleus with a tail. There's not much to it. There's no cytoplasm. It's just a nucleus that's surrounded by an outer plasma membrane that has a flagellum. And that's it. That's all sperm is. Now an egg, on the other hand, is this large cell, but the nucleus of that large cell is still the same size as that sperm. So what else is in that egg? What accounts for the volume of that egg? And it's not the nucleus, it's the cytoplasm. So an egg is mostly cytoplasm. Think of a chicken egg. We come across chicken eggs all the time, right? I'm sure everybody's eaten an egg at some time in their life. That chicken egg is all cytoplasm, right? It's not a fertilized egg. It has a single nucleus that's haploid. But the size of that nucleus is the same size as every other eukaryotic nucleus. It's so tiny you can't see it. But the rest of that egg is cytoplasm. What's a cytoplasm used for in an egg? Have you ever thought about that? The whole organism comes from that cytoplasm, right? So the beginning of embryogenesis, the beginning of that life, that new organism, comes from that cytoplasm. The nucleus is just instructions. And so all the proteins that are necessary to start the process going, to start that first mitosis, to start that second mitosis, all of those first divisions come from that first cytoplasm. And so the egg then is more important at this stage than the sperm. Does that make sense? And so we call this a maternal effect because whatever the mother has put in the egg is going to influence the early development of this offspring. Right? So whatever proteins are present are going to start the process. So we sort of try to think of this as an egg phenotype. So whatever proteins are in that egg are going to set that offspring going once the sperm enters, fertilizes, and the two nuclei combine together. So when we talk about maternal effect, it has everything to do with the egg and the environment that that egg develops in. Because especially in mammals, the mother supports the life of the offspring until birth. And so everything that goes into the mother's body is also going to the developing offspring. And so mothers have a tremendous effect, an environmental effect, and a phenotypic effect on their offspring. Think about that. If you ever heard of maternal diabetes, right? That has a huge effect on the offspring. It can actually cause either low birth weight or high birth weight, depending on which way when it happens. And it can cause problems in both the mother and in the fetus. Right? So when we think about our maternal effect, we're thinking about our egg. We're thinking about what's inside that egg. And so what is cytoplasm actually made of? Besides water, what's in the cytoplasm? Nutrients. So we got nutrients like sugars and oxygen and all that kind of stuff. What else? Anissa, you said something. Well, she said nutrients. Is that what you said? Mm-hmm. What else is in there? Ribosomes, absolutely. It's jelly-like. Why is it jelly-like? I'm sorry.
Yeah, permeation.
Permeation, is that what you said? Yeah, so transporters, right? And cytoplasm, thick. It's thick, it's not watery. What's in a eukaryotic cell besides a nucleus? I'm making you think. What makes it a eukaryotic cell? It's got
Membrane-bound organelles.
membrane-bound organelles. It's got an endoplasmic reticulum. It's got a cytoskeleton. It's got mitochondria. It's got proteins, enzymes, got glycolysis enzymes for sure. So it's full of all the things that a cell has, but it's all the things that the cell, that cell has produced. Okay. So we're talking about gene products in addition to nutrients. So whatever the maternal genome has expressed, whatever proteins have been expressed, whatever RNAs have been expressed, are out there in the cytoplasm. And it's really important that that's there because once this is fertilized, that nucleus does not have time to express its own proteins fast enough for the first division. So that first division comes fast like that. And so all of that stuff has to be in place for mitosis to happen. And what is mitosis? What do we need for mitosis? We need to condense our chromosomes, we need to split open the nucleus, and we need to assemble the mitotic spindle. All right? So all of those things have to happen using the proteins that are already there. Okay? So what does that have to do with whether a snail is left-handed or right-handed? Now, the handedness or which direction that snail turns, whether it goes this direction or this direction, you could call it counterclockwise and clockwise as well, either way you think about it. Our right-handed or our counterclockwise is also called dextral because dextral is the old Latin term for the right hand. Because at that time, most people were right-handed, and so if you were right-handed, you were dexterous. You know that word dexterous? And so it meant being able to use your hand efficiently, I guess. Now, dextral is typically wild type, right? Think about how many of the left-handed people you know. How many left-handed people do you know?
Myself.
Yourself.
And my mom.
And your mom.
And my
So
siblings.
obviously it's
My siblings.
inherited, right? Runs in families. Yeah. You probably, unless you're in a family of left-handers, you probably only know a handful, right? Although in college, it's kind of a selecting effect because left-handers tend to have better logic processing functions. It's related to that. And so we tend to see more left-handers in colleges and graduate programs. So, you know, it sounds like hocus pocus, but it's actually true. So left-handedness is actually the unusual condition. It's not the common condition. Now, in human populations, left-handedness is common enough now. It's probably about 30 to 40% of the population now because people are expressing their left-handedness. But back when I was a kid, if you were left-handed, they made you become right-handed. And that's why a lot of doctors of my generation have terrible handwriting, right? That's a cliche because they're made to write with their non-dominant hand. Now, whether it's dextral, right-handed, or it's left-handed, which is called sinistral. Does that remind you of another word? Sinister, right? So it used to be thought that the left hand was evil. So if you shook hands with your left hand, you were up to something because that meant that your sword was drawn in your right hand. So we have our sinister hand, we have our dextral hand, and so whichever proteins in the egg are ready to set up that first mitosis, whichever proteins are there, is what forms the handedness of the snail. So if your spindle sets up in this direction, going in this direction, it becomes right-handed. But if it sets up in the opposite plane, it becomes left-handed. That's the first cell division. Sets up the whole entire body pattern. And we seem to see that also with humans. It comes from that really, really early egg. That's really cool. Now, however, this is a regular old genotype. It's a Mendelian trait in the mother, but the phenotype is expressed as part of the egg, but it's expressed at a time point at which the egg has already become the zygote. So we consider this to be the mother is creating the phenotype in the offspring. Did everybody get that? Mother genotype and her expressed proteins create the phenotype of the offspring. Now, if you were doing a regular Mendelian cross where you had two parents that were heterozygous, so it's a regular old monohybrid cross, you would expect from that cross of big D little d times big D little d, you would expect a 3-to-1 ratio, 3 right-handed to 1 left-handed. But in fact, what you actually get is all right-handed if the mother was right-handed, which the mother is if it's dominant by dominant. So even though it's like a monohybrid cross, you never see that recessive because the mother is dominant. Okay? So your whole generation, your F2 generation, does not give us a 3-to-1 ratio. It gives us a 100% ratio based on the mother's genotype. So no recessive in that F2 generation. So let's kind of follow this cross as we go through to look at the F2 generation, and then we're even going to extend it to another generation. So up here we have the parents, and what we're doing is another type of reciprocal cross where we have the mother is dominant over here. So that symbol up there stands for female, the symbol stands for male. So we have the mother that's dominant, the father is recessive. Mother is recessive, father is dominant. Now if you cross them, you should get all heterozygotes. All heterozygotes. Now, if this is a Mendelian trait, what would that give you in the next generation if they were all heterozygotes? What would you expect if right is dominant to left? All right-handed, because that's dominant, right? Now, with your female as dominant and your male as recessive, you do get that. You do get all heterozygotes that are all right-handed. But when you do the opposite cross where the female is recessive and the male is dominant, all of the offspring have the same genotype of heterozygote, but because the mother was recessive, they're all recessive. All right? They do not show the dominant trait, even though they have the dominant genotype. Now take that another generation, and we'll look at the maternal times the paternal. So this is heterozygote by heterozygote. Now these females are dominant. These females are dominant. Right? Even though they're left-handed recessive phenotype, they have a dominant genotype. So in the next generation, they're all dominant because all of the mothers were dominant. See that? And then if we extend it to the next generation, because we do have some recessive mothers, even though they have the dominant phenotype of right-handed, those recessive mothers can produce some recessive offspring. Well, all of their offspring will be recessive. And so you come back to your 3-to-1 ratio, but only in the F3 generation. And that's assuming random mating amongst all of these. Okay? And you can work out all the different crosses. You could have big D by big D, big D. You can have big D by big D, little d, etc. You can work all through those different crosses, and they'll all add up to give you that 3-to-1 ratio. All right? Does that make sense?
Yes.
Wow, you guys are so smart. Most classes are like, no, I don't get it. Yay! Alright. So, you just follow it through. And so when you answer questions about it in the homework, you just have to remember that you're looking at the mother's genotype to figure out the offspring's phenotype. Because it can get kind of confusing when you have to produce it yourself. Okay? Now let's look at a little bit at the molecular reason for this maternal effect. Alright, so this is the egg, and the egg is not alone, right? The egg is surrounded by a follicle, right? And so during ovulation, the follicle kind of matures and starts producing all of these proteins to get the female body ready for pregnancy. And so these follicles have something called nurse cells inside them that line the egg. And so those nurse cells are going to be producing lots of proteins. And if you're heterozygous, those nurse cells will produce big D proteins and little d proteins. Even though the egg itself is haploid, right? Because this is an egg. So no matter what genotype that is, it's going to carry some of each of these proteins. Now, it's going to load them up and it's going to transport them into the egg using a transport system, protein transport. And so we wind up with an egg that has both types of proteins if the mother is heterozygous. So we're talking about both mRNA and proteins themselves. So it could just be an mRNA that they send in so that the ribosomes inside the egg start producing protein as soon as they can. All right, so those maternal nurse cells have a big role in setting up these phenotypes. And so if we look at the different ideas of what could happen, so if the mother is big D, then the only proteins and mRNAs that can go into the egg are big D. So that means that the egg is phenotypically dominant, which means that the offspring is phenotypically dominant. For a heterozygote, you're going to get both types of proteins, but because that's phenotypically dominant again, that means that the offspring will be phenotypically dominant. But if you have a recessive mother, there's no dominant protein being put in, so that means that it's phenotypically recessive for a phenotypically recessive offspring. So then, right after fertilization, right after the sperm nucleus and the egg nucleus fuse together to create that diploid nucleus, that zygote, we have them already making proteins and setting up the cell cycle to happen really quickly. It takes about maybe 12 hours for that first division to happen, which is not enough time for that newly fused nucleus to express a gene, because it can take days to make an RNA, to express a gene and then express a protein. Okay? There's not enough time before that first division, so it has to work with what it actually has. So the axis is set with that first division to make sure that every division after that goes in the same direction. So as the snail grows, it's permanently set on a specific axis, and it keeps growing that way over its whole lifespan. Do you guys know how snails grow? Yeah, they just kind of, as they grow bigger, their shell kind of grows longer and longer and longer until they're like a maximum size. So it's too heavy to carry around anymore, I guess. All right. All right, so that's maternal effect. There aren't actually a lot of examples of maternal effect that are as easily worked out as this one and as easy to see. We do know that a lot of very early developmental genes in humans have a maternal effect, but we haven't been able to study them as well. We just kind of go, well, it can't be anything else, so it has to be maternal effect, right? If you can't find the gene responsible, you kind of start looking for all of these other possibilities. Okay? There's one more possible phenotypic scheme that is available in your research. So if your Mendelian inheritance doesn't make sense, start looking for other things. All right. Now another thing that can cause differences in a phenotype kind of compared to what genotype you actually have is something called an epigenetic change. And epigenetics is a huge blossoming field. It just means that it has to do with chromosomes, but it's not necessarily a mutation or a change in the gene. So it's chromosomal, but it's not genetic, if that makes sense. So this becomes then a difference in how a gene is used or how a gene is expressed rather than whether the gene is expressed or not. This is kind of a when and where the gene is actually expressed. So it's not a mutation, but it is a change in expression. And usually reversible. So even if it's a chemical change, it means that that chemical change can be reversed given the right situations, because something put it on there, so something can take it off. And we actually see this a lot with changes in the environment. For example, with global warming, we see a lot of epigenetic changes in response to global warming in a lot of communities, right? And here I'm talking about like plant communities and insect communities. In fact, Dr. George Jex's research has to do with epigenetics in frogs, right? Has anybody ever heard him talk about his research?
Yeah.
Out in Arizona, right?
Oh, yeah.
Yeah, and it has to do with frogs responding through epigenetic changes to climate change. So this is another way that you can have short-term adaptations that aren't evolution. So this is not evolution, this is just changing the way the genome is used. So we need to think about what a chromosome is before we can really understand how chromosomes can change. So if you think about a chromosome in a eukaryotic cell, it's made of a single double helix, but that double helix has to be wrapped around these little protein discs called histones. And these histones can then kind of pack a little bit tighter together to create something called a 30 nanometer fiber. And that fiber can then be looped inside the nucleus to kind of pack it in there. And it's actually attached inside the nucleus where the loops are. And so when you think about chromatin, you can think about it as being either open and accessible, because if you have these little stretches of naked DNA, your proteins can get in there and use them, whether it's for replication or transcription or whatever. But if it's packed up like this, it's closed. So you have open versus closed chromatin. So you can control access to DNA, which is not changing the DNA in any way. You can control access by either closing DNA that was open or opening DNA that was closed. So say that you're a fish that lives in an area where the pond freezes over. And so when you go into wintertime, you may want to open up the part of your chromatin that expresses your antifreeze genes. And so you express your antifreeze protein just for the winter time because you don't need it in the summer. And then when the winter time is over and you're going back into spring, you can close it back down. So that's an epigenetic change in how that gene is used. Now, our genome is full of those epigenetic switches. We have a lot of switches, especially between seasons. For example, we're in the fall right now, and you're going to start, as the weather starts getting colder, you're going to start getting really, really tired, besides school, right? Besides studying and all that stuff, because your body has just opened up and ramped up production of your red blood cells. Why would your body want to make more red blood cells for winter? Picking up your blood. Keeps you warm, right? And that's a natural human reaction to the change in seasons. As the light changes, our bodies take that cue, that environmental cue, to open up and start producing more hemoglobin and more red blood cell proteins so that we can keep ourselves warm in the wintertime. Now, the problem comes when that happens not due to our environmental cues or a normal response to something going on. You can have maladjusted chromatin changes. And when that happens, you go into a disease state, right? So those of you going into medicine, this is a huge part of what you're going to be dealing with. Because how genes are used and where genes are used in your body can mean the difference between health and illness. If you think about, there's a condition called, what is that called? Heterochromia. Heterochromia, that doesn't sound quite right. What I'm thinking of is heterochromatosis, that's it. Heterochromatosis is a condition where your body stores too much iron. And what would happen if your body stores too much iron? What is iron? It's in your blood. You use it for hemoglobin. What else do we know about iron? It's ferrous sulfate, yeah, so you may have an ionized form of iron. What is an ionized form of iron? What is its main property? It's reactive, it's magnetic. So if you have this condition, heterochromatosis, and you have too much iron in your blood, and you hold a compass, it's always going to point to you. You'll get lost in the woods because you'll think you're always going, you're always going south. It's going to point to you. Heterochromatosis also causes a condition called Restless Leg Syndrome. Does anybody have that? I have it for a different reason, but it is annoying and you can't sleep. It's really annoying. Your legs just feel like there are ants crawling in them, and you just gotta shuffle them off. So creepy. Right. Anyway, you want to make sure that your chromatin is open for the right reason and closed for the right reason. All right. So again, we have our open chromatin. So if we think about genes, so for a gene you have to have access to the promoter to get it started, or if you need to do DNA replication, you have to have access to the origin of replication to get it started. And so if you keep those in these stretches of naked DNA between your histones, you have access to them. But if you wind them up, then you lose access and you lose the ability to do transcription or any other thing. Now here, the white circles represent cytosines in the DNA that are free and clear and ready for use. And here they label them as unmethylated cytosines. In closed chromatin, we have methylated cytosines. Does everybody know what I mean by cytosine? ACGT. So you can stick a methyl group. What's a methyl group? Organic chemistry students? CH3. Simple, the smallest organic molecule, right? You can stick that onto your cytosine, and your cytosine becomes methylcytosine. And methylcytosine tells the cell, we're not open for business. And the other chemical change that can happen is the histones themselves can be changed, because the histones grab the DNA, because the histones are positive and the DNA is negative, so they have this nice ionic interaction between each other. And so if you interrupt that interaction by acetylating the histones, which an acetyl group has 2 carbons instead of 1, then that kind of loosens up the histone's grip on the DNA. Now, methyl groups have the opposite effect. If you methylate the histones or take the acetyl groups off, you can tighten up the chromatin again. So this is two different ways of adding chemicals or subtracting chemicals that the cell can control chromatin. So there are enzymes that just monitor these areas of chromatin and do this. They acetylate, or they deacetylate, or they methylate, or they demethylate to control these levels. Now, where do these methyl groups and acetyl groups come from? How do you get things into your body? They come in your diet. And so some diets are high in methyl groups, and some diets are high in acetyl groups. And so if you have a diet that's high in methyl groups, then you have a lot of material to close down your chromatin. And that's why diet is actually associated with epigenetic changes. Also, pollution. Also, a lot of things that aren't good for us are associated with closing down genes. So, that's why diet is important. That's why making sure that you don't have too many of these donors is important. Mental donors. All right, so mammals also have an interesting phenomenon of epigenetic modification that is normal. And that normal epigenetic modification has to do with something called dosage compensation. So dosage tells us that, you know, the higher the dose, the more effect, the lower the dose, the less effect. And so when you think about the chromosomal difference between males and females, females have 2 X chromosomes, males have 1. Well, in females, 2 X chromosomes may be too much because a male can survive with 1 X chromosome. So to compensate for that difference and to make females not like twice as female, I guess, I don't know what that would actually do, but to make sure that they're compensated for, mammals tend to inactivate one of their X chromosomes in females, so that each female and male both have only 1 active X chromosome. Now the process of this is very involved and very complex, and we won't go through that now, but a lot of different species use this XX versus XY or XX versus X0 to manage their sexual ratios in their populations, right? Because you don't want too many females, because then you wouldn't have enough males to mate with them. You don't want too many males, because then you wouldn't have enough females to mate with them. So you want a good balance, about a 50-50 balance. All right, so if you close down one of those X chromosomes intentionally, then we will compensate for that problem, that balance problem. Now, in humans, we call that extra X chromosome a bar body. So here's a black and white image of a fluorescent microscopy or micrograph. And the X chromosomes, both X chromosomes are stained yellow in a background of red. And so here you can kind of see one X chromosome that's wide open, because it's kind of a diffuse yellow. And here you can see the one that's closed, the bright yellow. It's brighter yellow because it's compact. Now, how this happens is we go from open chromatin to closed chromatin. Now, actually, it's an even further condensation than that. It's more like a mitotic chromosome that it becomes. So how that happens is we have our original zygote receives 1 X from dad, 1 X from mom. Early in embryogenesis, say maybe after 4 or 6 divisions, it's very early on, one of these X chromosomes is randomly inactivated in each of the cells. So here's a cell that inactivated the blue chromosome. Here's a cell that inactivated the purple chromosome. And so every cell that comes from this cell is now going to have a purple chromosome inactivated, and every cell that comes from this cell through mitosis is going to have the blue one inactivated. So what you wind up with is a female organism that's patchy. Some of the cells have 1 X chromosome inactivated, some of the cells have the other one inactivated. So that means that if you're heterozygous, you may be recessive in some cells, and you may be dominant in other cells. And if you think about how many genes are on that X chromosome, there's a lot of possibility there for you to have patches of cells in your body that have 2 different phenotypes. It's kind of cool. Now, the places that we usually see this the best are in skin phenotypes, because you can obviously see skin. there's a human phenotype that has to do with the density of sweat glands. It's called anhidrosis. And so you would have a patchiness if you're heterozygote. You have patchiness where you don't sweat and you sweat a lot. So you're either dominant or recessive in some parts of your body. And if you happen to be dominant in like places that sweat a lot, that's kind of unfortunate. And that's called unfortunate lionization. Lionization is another word for bar body formation. Now this is an example in a mouse. So has anybody ever seen, they're called fancy mice, spotted mice? I mean, usually we think of them as either all brown or all white. Spotted mice are female mice, and there's a gene on the X chromosome that's either pigmented or non-pigmented, so black spot or white. And what you can see is the pattern of X inactivation. So the pattern where the mouse is white is where the recessive allele is on the open X chromosome. And where you see the black patches is where the recessive allele is inactivated. So the dominant allele is active. So you have patches in the body, and it's random. So you may have a spotted mouse that's mostly black with just a few white spots, or you may have a mouse that's mostly white with black spots. It's completely random how it turns out. Now another example in mammals would be calico cats, right? A calico cat is a female cat because the gene for pigment formation is on the X chromosome in cats. Not in us, not in other species, but for cats. So you either have the brown or black, and you have the orange. And so if you have a cat that has black and orange patches on it, that means it has either the black allele inactivated or the orange allele inactivated. You can rarely find a male calico cat, but that means he has to have 2 X chromosomes. So there is a way that you can determine how many X chromosomes someone has, and that's just by counting the number of X chromosomes they have, or counting the number of bar bodies they have. So if you look at the chromosome composition of the sex chromosomes, normal females have 2 X's, that means they have 1 bar body. Normal males are an X and a Y, that means they have 0 bar bodies, or 0 inactive X's. For someone who has Turner syndrome, which just means they only have 1 X, they have no bar bodies. So you can have a female with no bar bodies. However, if you have triple X, you have 2 extra X's, so you're going to have 2 inactive. Each cell will have 2 bar bodies. And then if you have Klinefelter syndrome, which is where you have 2 X's and a Y, they're still phenotypically male because they have that Y chromosome, but they also have a bar body. Okay? And these are the 3 most common chromosomal disorders among humans. You probably know someone who has one of these, but they don't know it themselves, right? Because they don't really produce phenotypes. They just produce uneven numbers of sex chromosomes. Right? Now, some genes on that X chromosome, on that bar body, some genes are never condensed, and that's just the gene that maintains the bar body, right? So you have to keep it going and make sure that it keeps going. All right, and that's it for X chromosome inactivation. Questions? We went over maternal effect, we went over epigenetics and X chromosome inactivation today, and so we'll finish up on Friday. One thing I did notice in the schedule, and I apologize for keeping you a minute over, I did notice that I think the 3rd test is the Monday after homecoming weekend, so I'm proposing that we move it to the Wednesday. Would you rather move it to the Wednesday after or the Friday before? I think there's stuff going on Friday. So let's move it to the Wednesday after. Yeah. So it'll basically be kind of a day of whatever, I don't know. Because I forgot to put fall break in the calendar too, so we'll just do what we were supposed to do then. Questions, comments, anything else? October 8th and 9th? Oh, yeah, you're right.
Oh, I bet this.
The 8th is a Friday.
Change it really quick.
11th and 12th, yes, you're right.
Oh my God. When we have the one that we left in class.
So the one so it's either or. So you either do the document or you do the assignment. So there's a pre-lab, there's an assignment, and then there's a, yeah, that's it. There should be 2 assignments for you.
The quiz and the assignment.
The upload is only if you didn't do the assignment. Because some students like to do the document instead of the assignment.
Oh, yeah.
So it's either or.
Well, I did both.
Good for you.
But I'm not doing it for
Okay.
Yeah, because that's right.